Okay, so lineage commitment is a topic that is still being investigated and is still under debate. So it's a topic that I chose to talk about today. Sorry. So lineage commitment is the development of a cell that can give rise to multiple cell types, but it's development of that cell into one of those cell types that it has the potential to develop into. So the best way to think about lineage commitment to understand this is kind of like a cell's pathway, and if it chooses to take a different pathway, it's going to end up at a different location. That's the easiest way. So these are both two different types of examples of lineage commitments in cells, and that's faced in T cells and then hematopoiesis. So in hematopoietic cells, their investigators discovered that the Notch signaling from the Notch receptor was key to inducing the development of T cell or B cell lymphocyte lineage. So when Notch1 is overexpressed, as you can see, in a hematopoietic cell, then T cells rather than B cells are going to develop in the bone marrow. But when a Notch1 gene is knocked out of a hematopoietic cell, then B cells rather than T cells are going to develop. And that's kind of an overview of the hematopoietic lineage commitment. Now in T cells, there's going to be different lineage commitments that need to be taken, that will take place. And there's a lot of different models that will represent that. So lineage commitment requires changes in the genomic organization and gene expressions. And these changes need to result, they have two things: silencing of one coreceptor gene, which is the silencing of the CD4 or the CD8, as well as the expression of genes associated with a specific lineage function. Cellular and molecular mechanisms responsible for lineage commitment are still under debate. That's why there's certain different models that are going to represent this. So we're going to be discussing the most recent perspectives. Several models have been proposed to explain lineage commitment: the instructive model, the stochastic model, and the kinetic model, or the kinetic signaling model, sorry. So first, the instructive model is basically the right engagement happening with the right signal. There's no apoptosis that's going to take place in this model. This is just a simple outline. So the TCR and CD4 and the TCR and CD8 co-engagement in this model is said to generate a unique signal which directly initiates the distinct development program. That's kind of like an overview of this whole thing. So if the thymocyte randomly generates a TCR with affinity for an MHC class 1, then the TCR and the CD8 would bind to the MHC class 1 molecule together, and they will generate a signal which specifically initiates a program that silences the CD4, so you'll have a low CD4, and induces the expression of the gene specifically for cytotoxic T cell lineage function. So it'll create the single positive CD8 T cells. Likewise, if the TCR and CD4 co-engagement generates a signal which initiates the CD8 silencing, so it would have low CD8, and it would induce the helper T cell development program, and you'd have the single positive CD4 T cells. So the next model is a stochastic model. No, sorry, the instructive model is kind of too simple and not really accurate. The stochastic model is going to be a little bit more accurate and a little bit more in depth because it's not the perfect environment, the perfect scenario. So the next model is a stochastic model. It entails the downregulation of CD4 or CD8 is random. So it's basically saying that randomly high or low levels of CD4 and CD8 are going to be expressed. And it's basically saying that only the cells that express the correct coreceptor, which are basically the cells that co-engage with MHC and TCR, are the cells that are going to receive a signal strong enough to survive until they're mature. So in the first line, you can see we've got random signals going in of CD4 high or low. And if the cell is able to bind from this signal, if the cell is able to bind to the antigen and the MHC class 1 molecule, then the CD8 cell is created. If it's not able to bind to both of those, then apoptosis takes place. You can see in this next line, we've got the random signals of CD8, high or low, going in. And if the cell is able to bind to the antigen and the MHC class 2 molecule, then the CD4 cell is created. If it's not able to bind to both of those, then that cell also undergoes apoptosis. So this pathway is basically asking if the cell's getting the signal, and if it's interacting with that signal, then it's going to let it live. The final model that we'll be discussing is the kinetic signaling model, which this model discusses or kind of represents this: if the thymocyte is to commit to the CD4 T cell lineage, then it must be receiving a continuous signal. But if that signal is interrupted, the TCR signal is interrupted, then it's going to commit to the CD8 cell lineage. So basically, we know that positive selection causes the downregulation of CD8 in all thymocytes. So cells that bind with the MHC class 2 are not going to be affected by the downregulation of this, and they will have a continuous signal from the TCR. So they will automatically develop into the single positive CD4 lineage. But the downregulation of CD8 does affect the signal with the MHC class 1 bound cells, and that sends the cells down the CD8 lineage. And that's very important in this. And the kinetic signaling and the stochastic signaling are both kind of more accurate than the instructive model, but each one separately is still not 100% accurate. And that's why these are still being kind of discussed and debated. But there are also other factors that are involved in lineage commitment, and these transitional factors are two examples. The THPOK and the RUNX3. They act by suppressing genes involved in the differentiation of lineages, and you can see that this explains how both of them inhibits the expression of certain genes, which then provides or forces the cell to take a different lineage or a different pathway, if you'd like to look at it like that. And there's also small populations of double positive thymocytes that can also commit to other T cell types. And these other T cell types that these ones can commit to are natural killer T cells, regulatory T cells, intraepithelial lymphocyte lineages. So basically all of this is still under debate and remains a topic of interest.